Good afternoon. Good evening, Sarma.
Good evening, sir.
Please confirm whether my screen is visible. Let me share my screen. So is it visible?
Yes, sir.
Okay. Thank you so much. So welcome everyone. Today we will be discussing about the week 10 assignment, and it is basically about the protein macromolecule interactions one, in which we will study the protein carbohydrate interaction, protein DNA interaction, protein RNA interaction. Then also we will talk about more and more things, which I hope you all have gone through the lectures that have been already uploaded in the NPTEL portal. So we will go to the next session, next objective of our talk. The first thing that I would like to revise you the brief concept that have been covered. These are protein carbohydrate interaction, carbohydrate recognition domain, then uh, then protein RNA interaction, then RNA footprinting assay, various assay which in which you might have studied how to detect the protein ligand complex or the protein ligand herein is nothing but the carbohydrate, RNA or DNA, which are nothing but nucleic acids. Then you must have studied about the basic DNA and RNA structure, then detection methods for techniques of protein nucleic acid interaction. The important keywords that you might have come across are glycoconjugates, proteoglycans, then glycoso glycosaminoglycans, glycoproteins, then phosphorylated saccharides, their functional roles, their importance in the biological network or in biological organism, then transcription factors about histones, what are the roles of histones and all, then the DNA binding domains, RNA binding domains, RNA recognition motifs, then dead box domain. These all terminology you might have come across. Okay, so the contents is as usual. First, we will discuss about the practice questions on this particular current module that you might have gone through. Then the discussion on important topics. Then I will take some doubts from you if you have any. And then we will take the scope of this module with research perspective. Now, coming to the next slide. Uh, First, your first question is related to the glycosylation. You need to identify glycosylation is defined as whether the carbohydrate is attached to protein or lipid or to a metal ions or to a peptide. What do you think?
Option A.
Option A means carbohydrate attached to a protein. Very good. This is a correct answer. Glycosylation is defined as the in this glycosylation process, mostly the carbohydrates are being attached to the protein. Okay. So next, your next question is which proteoglycan is critical component for cartilage structure and function of joints? You might have studied various proteoglycan in your lecture 46, lecture number 46. You have ma'am has discussed about various proteoglycans in which she talked about various types of them and their critical role and their like their role and function in the human body or in mammalian cells. So your options are agrine, then agricane, precline, uh, perlecan, or lectin. So which of the proteoglycan having the critical component for cartilage structure and function of joints? Can you recall which among these proteoglycan has this critical function? Any idea? Your correct answer is aggrecan. So aggrecan is a type of proteoglycan which has the critical component for cartilage structure, which are found when the two joints are which happen to be playing an important role where two bones are getting joined. So there, this particular proteoglycan plays an important role. Now coming to the next question, question number 3, that you need to identify the lectins. Here, these are represented by X, Y, and Z, 3 letters. You need to identify which type of lectins are for the different role. The options are here. The question is X type lectins are essential for cell adhesion. Y type lectins are essential for development and maintenance of nervous system, and Z type lectins essential for the role in generation of functional rhizosome. Your options are X stands for I type, Y stands for P type, Z stands for C type. Similarly, B, X stands for P type, Y stands for I type, Z stands for C type. In option C, X stands for P type, Y stands for A type, Z stands for I type. And your option D is X stands for C type, Y stands for P type, and Z stands for I type. Can you recall? There are 3 types of lectin, right? You might have come across that in your lecture number 46 itself, I guess. Lecture number 46 or 47, somewhere in between that, where ma'am has taught you about the protein carbohydrate interaction. Protein carbohydrate interaction, where ma'am taught you there are 3 type of lectins with figures also, that their crystal structure. So can you identify which type of lectin is essential for cell adhesion? Then it will be easy for you to filter out the options. Any guess? Basically here cell adhesions are facilitated by C-type lectins. So if you remember that particular answer only, then you can filter out. The correct answer here is D, because here the C-type lectins, that because they are essential for cell adhesion, lectins are nothing but they plays an important role in adhesive. They act as adhesive molecules. Okay, so here here C-type lectins are essential for cell adhesion. However, for the development and maintenance of nervous system, you require P-type lectins. Okay, and Z-type lectins are essential for the role of generation of functional lysosome, which act as a agent for autophagy. Can somebody unmute themselves and tell me what is autophagy?
Self-lysis of a cell.
Self-destruction of the cell. Very good. So here the lysosome plays a lysosome is also known as the suicidal bag. And in that suicidal bag, which plays an important role in autophagy, the I-type lectins are mostly involved. And these are involved in the generation of functional lysosome. So your correct answer here is D. Okay, correct answer is D. Now coming to your next question. Histones are composed of which all histone proteins? Can you recall? Histone. First tell me what are histones? Can anybody mention what are histones? Yes.
That supercoils the DNA into chromosomes.
Yes, these play important role in the coiling of DNA, right? Because of them only, they like the DNA double extended DNA get coiled up. Okay, so can you tell me for histones how many which all particularly are important and which all subunits are involved in the formation of histones? Your options are your options are Your options are H2A, H2B, H3, H4, or H1, H2, H3, H4, or H2A, H2B, H3A, H3B, or H1, H2A, H2B, H3. Can you recall which are the component for histone?
Sir, option A.
Option A. Very good. Option E is correct answer for this question. Here the 4 subunits because histones are octamer. In octamer, these are the two replicating two similar discs are being joined on the beads like H2A, H2B, and H3, H4. Okay, these beads are divided something like this, where somewhere the H2A, H2B, H3, H4 are being attached. However, the H1 histone proteins are found on the beads, I guess. I don't remember correctly, but the H2A, H2B, H3, and H4 are essential for making the disc. And this one disc and second disc are coiled up, are stacked about them stacked, and then the DNA is coiled up in like this fashion around the histone protein. Okay, so the correct answer is option A. Now coming to your next question, question number 5. Among which of the following is not true? You need to find out the false statement about the mechanism of transport through the cell membrane. Okay, which of the following is not true about the mechanism of transport through the cell membrane? Is the correct question? Okay, yeah. Your options are: option A is DNA replication, option B is RNA processing, option C is role in the regulation of chromatin structure, and option D is oxygen transport. Which of the following does not transport through the membrane?
The DNA replication.
DNA replication. Do you think?
Actually, the question is not clear. Is it asking for the proteins that is being transported?
Yes, for the process which is not facilitated through the cell membrane, which are not being transported from cell membranes. For this particular question, the question is like that. If you think here, like the all the role the regulation of chromatin structure, this needs the protein to be transported from the cell membrane to the nuclear, in the nuclear cell, right? Because the regulation of chromatin structure happens in the nucleus. RNA processing can occur in both in nucleus or in cytoplasm, right? And DNA replication occurs in nucleus, right? So if you collectively remember, nucleus also have a membrane through which the proteins need to be transported. So in all three, the cell membrane transport, the transport through cell membranes is being involved. However, the oxygen transport only occurs at the surface because hemoglobins are the transporter molecules which transport oxygen from one place to another place. In this process, the oxygen transport does not require the cell membrane transport through the cell membrane, right? So here, your correct answer here is oxygen transport. It does not it is not true about the mechanism through the transport of the cell membrane, right? Okay, so now the next question is the sugar phosphate backbone. Sugar phosphate backbone. Where does it find? Where do you find the sugar phosphate backbone? Can you recall? Can you please in the structure, backbone structure of DNA and RNA, right? Nucleic acids. Now your question is the sugar phosphate backbone can interact with the proteins through which mechanism or which interaction? Options are van der Waals and stacking interaction. Option B is covalent and stacking interaction. Option C is electrostatic and static interaction. Or option D, van der Waals and covalent interaction. Can you guess? Sugar phosphate backbone can interact with proteins through which type of interactions?
So I'm guessing, is it option D?
Option D. Why do you think?
Sir, the stacking interaction will take place with the bases of the DNA and the protein. And in the electrostatic interactions is going to be take place with the the electrostatic interaction is actually in pair with stacking interaction. That's why I am neglecting this.
No, but actually sugar phosphate backbone, which charge it has? Sugar phosphate backbone?
Negative charge.
Negative charge. So obviously the electrostatic interaction will be there because the question is can interact, right?
Yes, sir.
Because backbone has negative charge, so electrostatic interaction will be there, and stacking interaction can also be there, as you explained, right? Due to the presence of metal ions.
Yes. Okay.
Right.
Then option C.
Option C is correct here. However, van der Waals interactions are kind of non-covalent interaction, right? So van der Waals interactions are less to be these are less possible to be appear when the sugar phosphate backbones are interacting with proteins. Can you please recall which all motifs can interact with sugar phosphate backbone? These are mostly will be found in the here in the major group or minor group, right? So which all motifs can interact with this? Can you recall in DNA? Those are the zinc finger, right? You must have studied zinc finger.
Magnesium ion containing motifs.
Yes. Zinc finger ion, zinc finger motif. Then another motif will be helix, helix turn helix.
Helix turn helix and helix loop helix also.
Helix loop helix. All three can interact sugar phosphate backbone, and those are being interacted with the formation of electrostatic and static interaction. Okay. Now the next question is replication protein A RPA, right? Shows much higher affinity for single stranded DNA, RNA and single stranded DNA, double stranded DNA, or RNA. Replication protein A.
Option A.
Option A.
Yes.
Are you sure? By which mechanism does it make the higher affinity? Why it is having the higher affinity?
Sir, actually it stabilizes the single-stranded DNA so that it doesn't get degraded.
Yes, very good. Because normally DNA exists in the double-stranded form. However, when the unzipping occurs to form the replication, when the one strand is being replicated, so in that case, the single-stranded DNA are being formed. So in that case, the single-stranded DNA are more probable to being degraded by the DNase enzyme. So to increase its stability, the replication protein A, RPA, are being bind to the single stranded DNA so that it provides its stability and therefore it has the higher affinity towards the single stranded DNA. Right? So option A is correct. Very good. Now the next question, the DNA binding domain of the leucine zipper domains primarily contains which type of amino acid? DNA binding domain. Okay. We are talking about the DBP. DNA binding domain. So here in this DVD, DNA binding domain of the leucine zipper. Leucine zipper is nothing but the motif, right? Nucleic acid binding motif. Or so here, if you recall, can you please tell me which amino acids plays an important role in leucine zipper? Your options are alanine and histidine. Option B, glycine and proline. Option C, arginine and lysine. Option D, leucine and alanine. What is the charge on leucine zipper? Positive or negative?
It should be positive.
Positive charge because it is binding to the negatively charged major group, right?
Then it should contain arginine and lysine.
Very good. And so that is why I asked. So this is how you can this is how you can fragment the question and try to understand. If you don't if you don't know the factual situation, in the factual situation when the question is asked as a factual question, so in that case what you can do, you can try to fragment the question and try to think about what kind of the leucine zipper will what kind of charge will leucine zipper have. So in that scenario, you will be able to answer more accurately, or like you will be able to predict the question, predict the answer. So however, if you remember, the leucine zipper is a alpha helical protein, right? Both of the zip is contained on the alpha helix, right? They are formed like this. So glycine and proline, these are known as a alpha helix breaker. So obviously this option will be incorrect, right? Because if you remember in your lecture one, lecture one in the week one module where I had discussed about the assignment related to the where I discussed the basic property of amino acids, the what are the amino acid property and where they can be found. So in that I had told you that glycine and proline, these are the alpha helix breaker. These cannot be found in the alpha helix. So obviously the glycine and proline will not be the correct answer. Here alanine and histidine, that might confuse you, but also alanine is a small molecule, a small amino acid. So it also has the less probability to be found in the alpha helix. However, if you deduce that the leucine zipper has the positive charge, so the arginine and lysine are the only amino acid which having the positive amino acid in this particular what you say options. So option C is correct in this question, right? So the correct answer for 7th was single stranded DNA. Here option C. Now the next question. The next question is, which of the following technique is used to accurately determine the DNA protein association constant? DNA protein association dissociation constant. So your options are site directed mutagenesis. These are the biotechniques to accurately determine the DNA protein dissociation. You need to find out which of the technique can be used. Option A is site directed mutagenesis, SDM. Option B, polymerase chain reaction, PCR. Option C, DNA footprinting. Option D, HDS page electrophoresis. Can you tell? What could be the possible answer for this?
Option A.
Site-directed mutagenesis. Can you recall what happens in site-directed mutagenesis, SDM?
I
What
know
do
the rest
you?
three.
Oh, you know the rest three.
Yes.
So let's start the like elimination technique. What is related to the polymerase chain reaction? What do you do
So
in
in,
polymerase chain reaction?
so in polymerase chain reaction, we keep on replicating the DNA up to 30 cycles.
Okay. Basically, we do for when we need to when we
And
have a DNA
amplify the
sample.
fragment of DNA.
Uh, amplify the fragment of DNA. We use the polymerase chain reaction. So in that this is nothing to do with the identifying the DNA protein dissociation constant, right? So option B is incorrect. Now coming to the Hts page electrophoresis, which is the most common. So what do you do in Hts page electrophoresis?
SDS page electrophoresis is done to analyze the molecular weight of a protein.
Protein, very good. So or to identify the protein, right? So
Yes, yes, sir.
this option is also incorrect. Now you have two options: site-directed mutagenesis and DNA footprinting. Now,
DNA footprinting, sir, we actually can mark where the protein is going to get bind in the particular sequence.
yes, and also this is kind of a macroarray assay, right? In DNA footprinting, what do? You like label the DNA and then like and then you test where it will going to bind.
Yes,
So
sir.
normally this involves the DNA protein interaction, right? However, site directed mutagenesis, if you don't remember, if you don't know what is site directed mutagenesis, uh, okay. In site directed mutagenesis, what happens is like you mutate a particular amino acid and then you check whether this having the particular significance role in altering the function of protein. Okay, so as the name suggests, site-directed mutagenesis, you know particular site and then you mutate that particular amino acid and then you check whether it has a role in altering the function of protein. So these all three, like these two are related to the proteins and this particular is related to the amplifying the particular DNA strand. However, when you bind the DNA and in that particular thing when you flow when you rinse the particular cell and then you check whether it is having a dissociation constant like so basically DNA footprinting assay is the only technique which tell you among these four the DNA footprinting technique is the only technique which tells you about the DNA protein dissociation constant means which involves the DNA protein interaction, then only you will be able to find out where the particular DNA strand will bind in the protein, right? So this is only telling about the binding of the DNA in protein. So if you know the binding of DNA, then you can determine the dissociation constant. Okay, so your correct answer here is C, not A, because A is related to the site protein to alter the protein structure. Okay, clear?
Yes, sir.
Okay, so now next question is which ribonuclease cleaves RNA in the middle of the strand that is an endonuclease. Okay, so your options are ribonuclease Z, ribonuclease P, ribonuclease A, or ribonuclease 3. Which of these are the endonuclease? Can you identify? What are endonuclease?
The endonuclease is cut any nucleotide sequence from middle of the sequence. Any
Yes.
generally in a palindromic sequence.
Okay, very good. And then can you identify RNA in the middle strand? Why does it cut in the middle strand? Which of these cuts in the middle strand? First, find out that. This is a factual question. Actually, you need to remember this. Actually, these questions, this particular question have been asked in many competitive exams. So if you know this, and then you will just take and move ahead with the another question. So that is why I have included this in the assignment practice. Normally, ribonuclease A are the endonuclease which cleaves the RNA in the middle of the strand. Okay, so here option C was correct. Okay. Now the next question is histones interact through dash, making a dash with the dash of DNA. You have three statements and you need to identify. You need to make this statement correct. Your options are histone interacts through basic residue, making ionic bonds with acid sugar phosphate backbone, acidic sugar phosphate backbone, or histone interacts with acidic residues, making ionic bonds and with the nitrogenous base of the DNA, or histone interacts with acidic residues, making hydrogen bonds and with the nitrogen base of the DNA, or histone interacts with hydrophobic residues, interacts making a covalent bond and with the sugar phosphate backbone. So what is the correct answer, do you think?
Option A.
Option A.
A.
Okay, very good. Because histones are mostly containing the histidine amino acid residue. These are the basic. Okay, these are mostly comprised of the histone or and also similarly they also contains basic residues, basic amino acid residues. So they interact through the basic amino acid residue and makes ionic bonds, not the covalent bond. Okay, and these are if it is basic, then sugar phosphate backbone should be acidic in nature, right? So these interact with the acidic phosphate sugar backbone. So option A is correct. Okay. Now next question. Replication is this question repeating again? Oh, this question has been repeated again. Okay. So here the correct answer was a single stranded DNA. Okay. Next question. Dash bind to the lac repressor, causing an allosteric change in its shape. Lactose or glucose or allolactose or sucrose. What is the correct option? Lac repressor. 13. Do you remember the lac regulation?
The option
Lac
C.
regulation?
Sir,
Option C. Okay. Yeah. Actually, this is being like lactose, the analog of lactose, which is allolactose, and this act as a allosteric changes, like allosteric regulator of lac repressor gene. Okay, so option C is correct. Now, ribosome consists of one or more.
The option A.
Option A. Ribosome consists of one or more rRNA, mRNA, DNA, or tRNA. Option A. Can you justify your answer?
The rRNA stands itself for ribosomal RNA, whereas mRNA stands for the messenger RNA, which will be translated into a protein, and DNA is not present in any ribosome, and tRNA are the transporter molecules that are basically bring the amino acids. So permanent residents are rRNA.
Okay, so can you tell me which in mammalian cells, mammalian ribosome, like or in eukaryotic ribosomes, which all rRNA are being found to form the ribosomes?
The exact distribution is not right now I can't recall it.
So have you known to about 18S RNA, 16S RNA, something like
Yes,
that?
sir. Yes, sir. Yes,
23S.
sir.
Yes.
Yes,
So
sir.
those particular things, those RNA are being formed in the, and for bacterial cell, can you recall? Because that is a very
Yeah.
famous technique also.
Yes, sir. Well, that is 16S rRNA is the
16S
most
RNA.
important one.
Yes, yes. So ribosomes consist of a rRNA. Very good. Now the next question is, in the figure here, in the figure, RNA binding. Let's wait 1 minute. Give me a second. Somebody was calling. Okay, sorry for the interruption. Now let's again come to the question number 15. Here you have given a protein structure. Like here you have given a RNA binding domain. Shown in the figure is a type of which domain. Okay. Can you identify whether it is dead end box domain or S1 domain or K homology domain or S2 domain? This is a factual question. Can you identify?
Is it an S1 domain? I'm not sure.
No, actually, it is a K homology domain. It's not S1. Okay. The next question is, in this particular type, okay, in this particular type of DNA binding domain, this is a certain protein which have the given here the DNA binding domain. So in this DNA binding domain, which of this having the particular type of motif? Your options are helix turn helix, leucine
So
zipper,
option C.
helix loop helix, or option C, helix loop helix. Are you sure?
C, because it's not a turn and it's not a leucine zipper.
You again check. Let me zoom if it is clear. Yeah. Now can you see it? There are helix, involvement of helix, definitely, right? So here there is a helix, and then again there is a helix. So obviously it will be helix, but it is a turn or loop.
So I think it's a loop.
This loop?
Yes, because
No,
turns
actually
are shorter.
turns are shorter, but here in between here because they are binding in the major groups, right? So obviously loop helix is also binding into the like its major group region, but this particular figure is related to the helix turn helix domain. Okay.
Yes, sir.
So 16A. Now the next question is, here on the ribosome, the pathway of a tRNA during polypeptide elongation is here. Can you identify which all sites can be there?
The option A, the A side,
Option
then
A.
B side, then E side.
Very good. Can you explain what are A side, P side, and E
Uh,
side?
A site is the amino acid site where the tRNA first, the second tRNA is going to come. The first tRNA will bind to the P site, that is the peptidal site, and E is the exit site from where all the tRNA is going to leave the ribosome.
Can you mark here which all are the A, P, and E site?
So this violet or purple one is the A site, this green one is the P site, and this yellowish one is the E site.
Yes, very good. So option A is correct in this. Now, identify A and B from the absorption spectra. So you have given a absorption spectra, which is having a 260 nanometer, you are having a band, and 280 nanometer, you have having a band. You need to identify which particular spectra stands for which type of molecule, macromolecule. Option A.
So A is DNA and B is protein.
A is DNA and B is protein. Are you sure? So
yes.
option D. So can you tell me why DNA gives 260 nanometer peak?
That because of the aromatic groups present, basically the rings or the nitrogenous bases that gives maximum absorbance at this 260 nanometer.
Very good. And however, 280 nanometers are shown by
proteins show maximum absorption in the this range 280. However, it may vary. I have read somewhere. So,
yeah, it may vary.
Also, yes, 196 or there also it can give a maximum range, and also the DNA protein hybrid, it's also going to give a maximum absorption spectra at 260 nanometers.
Yes, yes, very good. So why protein gives at 280 nanometer?
So because of the aromatic tyrosine and the tryptophan, I think.
Yes, very good. So option D is correct in this question. Now your next question is, you have isolated a protein. Okay, you have isolated a protein. You have isolated a protein and expect that it will bind to a specific sequence of double-stranded DNA. Okay, you have performed DNA electrophoresis, EMSA, DNA electrophoresis, phoretic mobility shift assay using DNA fragment as probe and got the observation as shown in the image. Like these are the images. This is the image. What would be your conclusion from it? Okay. Option A. The protein bound DNA fragment has the same mobility as the unbound DNA fragment. Option B. The protein does not bind at all with the DNA fragment. Or option C. The protein actually does not bind specifically to the DNA fragment, but have a non-specific interaction with the DNA fragment. Or option D. The protein binds specifically with the single-stranded DNA and not with the double-stranded DNA. Can you identify? Here you have three lane A, B, and C lane. Positively charged is above and negative charge is above and positive charge is below. So you need to identify. You read the question again. Your question is, you have isolated a protein, okay, because you have run a Hts page. You have isolated a protein and expect it to be bind to a specific sequence of double-stranded DNA. So which all protein or motif can bind to the double-stranded DNA? Can you tell me? Which all proteins can bind to the double-stranded DNA? We have discussed a few, like in back few slides. Can you recall?
The S1, the leucine zippers, are you asking for these?
Yes, yes. Those are nothing but the protein. Motifs are also protein instances, right? So I was asking that one only. So those can bind to the, so obviously when the question says you have isolated protein and expect it to bind to the specific sequence of double stranded DNA, means they are kind of a motif. Now you have performed a DNA electrophoretic mobility assay, which is EMSA, and using a DNA fragment, that particular sequence that you want to bind as a probe, and got the observation shown in the image. What should be your conclusion from it? Here, the if you have one lane, another one is B lane, and another one is C lane. Now you check the protein bound to the DNA fragment has the same mobility as the unbound DNA fragment. Protein does not bind at all with the DNA fragment, or the protein actually does not bind specifically with the DNA fragment, but does have the non-specific interaction with DNA fragment, and the protein binds specifically with the single stranded DNA and not with the double stranded DNA. From this EMSA, can you tell me?
I'm guessing will it be option B?
Option
B.
B or D?
B means
D for dog.
no. B bad.
B.
I'm not sure about this because if it's a single-stranded DNA, then the major proteins that will bind are the enzymes like polymerases and all that.
Okay, let's assume that only you have the you have protein and that particular protein and DNA in your mixture. Now you are running the EMSA or electrophoretic mobility assay, and then you run a gel. You see which let us say this is a protein, this is a ladder. Then you have this particular lane. You have only only DNA, and this is DNA bound protein, and then you run. So you are seeing the both have moved to the same thing, like same height, same distance they have moved, right? Their drift are the same from the initial condition, or their molecular weight are same. You can infer two things, right? In Hts page, what do you infer? Like how far it has moved, the molecular weight will be higher or
Same.
lower?
So if they are moving in the same range, then they are having the same molecular weight.
Same molecular weight. Very good. Then what you can infer, if both of the bands have been at the same height,
Then they are having same molecular weight.
same molecular weight means whatever you have run is contain only protein, right?
The
Because
only protein.
if it is bound with DNA, then its molecular weight will increase. It will not
Yes,
have the
sir.
same molecular weight because they are at the same height. What you can infer? You can infer that these
There
are
is no
having the
change in molecular weight.
there is no change in molecular weight. That means this band contain only protein. Means the protein does not bind with the DNA fragment. The second lane is also having the same protein, only protein. That is why this is having the same height in the chain. So that is why the correct answer is B. The protein does not bind at all with the DNA fragment because it is having the same height. No change, no molecular weight change. Okay. So by this, you are done with this assignment. Okay, now coming to your next question, next section, which is related to the discuss the important topics. So before that, do you have any doubts related to related to this module? Any doubt? So till now we have talked about the basic structure of amino acids in module 1. Let me recall all the things through the website. What all topics we have covered so far. So we have covered amino acids, peptide bonds, then the protein architecture, in which we studied the what kind of what kind of structure it can have, or what kind of configuration it can have, what kind of bonds are present in that, what kind of angles that are present in the two amino acids when two amino acids are joined. Then you have studied the protein folding, where you studied the Anfinson experiment. Then various different techniques to, and the molecules that are ready for the readily available, that can degrade the or denaturate the protein folding. Okay, which can inhibit the protein folding and can help in the denaturation of the protein structure. Then week 4 we studied the protein isolation and characterization. Then protein ligand interaction, in which we studied about the various interaction, like and how to quantify that, the basic terminology related to those type of interaction. Then membrane protein we studied the transport phenomena, how the transport mechanism is occurring. Okay. Then the enzyme and enzyme mechanism, enzyme kinetics, where we studied the different kinetic models, the different mechanisms. Then metalloproteins of the motor proteins, which we studied in the uh, last module, where we studied about the what we studied, the motor proteins such as tubulin, the macrotubules, then dynein, kinesin, right? Then protein. In this module, we studied the I guess it is mixed. In this module, we are studying the protein carbohydrate interaction, right? So now, till now, do you have any doubt related to any of the any of the module? Any doubt? So important. I will tell you the important topics which will be essential for your exam, the in semester exam. So here you can expect the question from mostly from if there are subjective questions, that it will come from the enzyme kinetics and enzyme inhibition. So try to follow which kind of mechanisms are there and how these mechanisms are important for enzyme inhibition and enzyme kinetics. So then you can also study, like in last module, week 9. In week 9 we studied what did we study in week 9? In week 9 we studied
the motor
membrane
proteins.
no, in week 9 we actually that was shuffled actually. So here this is the correct layout, I guess. Here also it is not accurately depicted, but here in week 9 we studied the membrane proteins and the motor proteins, right? The and electron transport chain basically. So here in the transport phenomena, so you can expect the question from transport phenomena or electron transport chain. Okay, so these are the important topics that will be there in your competitive exam also. In week 8 you studied the metalloproteins and the about the different proteins such as that kinesin, dynein, motor proteins and microtubules, kinesin, right? So you can expect the question from myoglobin and hemoglobin, because this myoglobin and hemoglobin, you have been studying this from the so long, like from week 1 also you had the introduction about the myoglobin and hemoglobin, because these are the oxygen transporter enzymes, right? Then in the week 1 question, you will be getting the question related to about the protein architecture mostly, and the peptide bond, the types of peptide bond, then the angles that can occur in the peptide bond, that from there you can expect the question. Okay, so basically these are very important aspects of the protein biochemistry. Okay, then if the questions are coming from the thermodynamics, those are mostly related to the mechanism of thermodynamics. So keep focus on the thermodynamics of the protein folding, then the thermodynamics of protein unfolding, then what type of bonds are essential for the giving the protein a particular kind of structure. So these kind of studies you can follow. Then in the protein week 4, you can expect the question from the biophysical methods, in which you studied the different quantification techniques such as NMR spectroscopy. Then you studied the X-ray crystallography. Then you studied the GCMS. Then you studied the HPLC techniques. So those techniques you can recall. Okay. Then in week 5, what you studied the protein ligand interaction. So week 5, week 6, and week 7 are the mostly related to the numerical type of question you can expect from here. Numerical question you can also expect from the membrane transport, where we solved few kind of questions related to the like membrane potential, right? In membrane potential, I had solved few of the question in the like while I was discussing the problem, assignment problem also. So you can practice those kind of question. And week 10, as we discussed here, the questions you can guess which will be related to the what kind of geometry it has. Then you can, if the different phenomena which are there in the protein carbohydrate interaction and then protein DNA interaction, there you can find this kind of question. Okay, so this module is also very much essential for in with respect to the when you in research perspective also it is very much important, because what kind of research you can get in this module is like if you have a if you similarly one question I have given you here in the question number 19. In the question number 19, this was related to like this kind of question can also occur. You can also find out this kind of question in your CSIR net exam in the type C question. Type C questions are mostly related to the experimental techniques and the various biotechniques. So in that biotechnique section, you can find this particular type of question. So the lectins and all are also very much important topic as far as it is concerned with the CSIR net exam. Okay. Then these kind of question, these are the analogy based question. So these analogy based question you can also expect in the interview, where if you are appearing for the PhD interview, there also you can find this kind of question. Okay, so try to solve as much as possible, and you can follow a book by I hope you are following, like you know about the one Disha publication book is there, right? Disha publication book, I guess. But I actually I am not recalling the name correctly. I guess if it is given here. No, if it is not here, then the books are like, let me check on the website, website. You can follow those books also. Those are specifically related into the for cracking the competitive exams, biotechnique. Yeah, this book. No, not this one. One book is there. The publication name I is not recalling. Biotechnique book. Okay, I will find out and then I will send you. Okay. So you can follow that publication book, and then you can like prepare accordingly using those books, and then that will be very helpful for you to appear when you are getting when you are appearing in the GATE and CSI net exam. Okay, so on rest, like you can for biochemistry, ma'am's lectures are much more like more than enough if you follow and if you remember everything about it. So the lecture material is also there in the website, and try to submit the assignment on time. Okay. So if you have any other doubt, you can unmute yourself and ask. Is there any doubt, Sanva? Okay. So we are done with this today's session. So thank you so much for joining. I hope you will join in the next sessions also, and I will upload this in the YouTube as as I upload all the lectures, and you can find that in my YouTube channel if you want to revise it again. So thank you so much for joining. Let's meet in the next session. Thank you.